Welcome everyone to today's seminar. I'm glad to introduce Dr. John Hardy today. John completed his undergrad studies in chemistry at University of Bristol, followed by his PhD in group of Professor David Smith in the University of York, focused on biomaterials chemistry. He subsequently undertook a decade of interdisciplinary postdoctoral training, first as an Entente Cordiale Fellow in the lab of Nobel Laureate Professor Jean-Marie Lehn in France, father of supramolecular chemistry. Then as an Alexander von Humboldt Fellow in lab of Professor Thomas Schiebel in Germany. Then he did several postdocs after at Christine Schmidt Lab at University of Texas, and later University of Florida, and finally Industrial Research Fellow in the lab of Professor Colin McKay in Queen's University, Belfast. He began his independent research career as a lecturer in materials chemistry at Lancaster University in August 2015, and currently he's a senior lecturer at this university. So he developed basically the first examples of biodegradable electroactive polymer-based drug delivery devices and biomaterials with biomimetic properties for drug delivery, neuromodulation, and tissue engineering. Now he leads an interdisciplinary research group undertaking basic and applied research for bioelectronics for biomedical applications. He has more than 12 patent families, and more than 90 publications, several grants from UK RI, European Developmental Funding Industry, and several international funding. So thank you, John, for being today, and we are eager to have your talk on biomedical or bioelectronic biomaterials.
Super.
Thank you.
Thank you very much for the kind invitation. So can I just check that the microphone is working okay?
Yes.
Super, okay. So what I will show you over the course of the next 45 minutes or thereabouts is some of what's been published, some of what's under review, and a few things which are just about to be submitted. There's a few, because of the nature of this being online, then I'm a little bit cautious about putting out there the stuff where we haven't yet got the IP absolutely pinned down, or the publications are like 6 to 12 months away. But I'll show you some things which I think are hopefully kind of in interest to the broad audience which is here, which I understand is composed of physicists, engineers, chemists, biologists, and others. And so I hope that there will be something in this for all of you, in the same way that the opportunity to come to Spain meant I got the opportunity to see sunshine and eat food where the food had actually seen some sunshine. So the tomatoes tasted of something, the herbs also tasted of something, and that's always kind of a nice opportunity to come back to Spain. I'm going to put the timer on because I recognise that this is possibly just before lunch and everyone wants to get away and escape to have some actually good food as well. And then without further ado, I'll show you some of what we're up to at the moment. This image here of Grandpa Simpson is one you'll see towards the end of the talk. And so when you're beginning to flag and think, I'm desperate for a coffee, I'm desperate for my food, when you see Grandpa Simpson again, you know we're on the home run and it's the final few slides. So you don't need to worry too much about that. So if I understand how to use this correctly, which I probably don't, I thought I did, I'll use this now. In a moment ago this worked, and now it's... okay.
No? When you are doing the streaming, I think it doesn't work.
So how could I change the slides?
I think maybe you need to go...
You have to do it with the keyboard.
Okay.
Here.
So if I'm here now, could you confirm that you see the slide move on your screen? No.
I can't see it.
You can see it online.
I see the presentation.
No, but it doesn't move.
The same slide that we are watching.
Oh, did you just do something?
No.
With the keyboard, it doesn't work with the keyboard?
It doesn't
Nope.
work.
Where is the mouse?
I'm not sure.
There.
Now it's working.
Oh, is it on a separate screen? Okay.
So you can.
Okay. I'll use
Okay,
the
so
keyboard
maybe-
now. So can you just check that you can see that online as well?
And we go to the next.
Yeah? It's online as well?
Yes.
Awesome. Great. Okay. So apologies for that. Hopefully the rest of it will run without any further complications. And then I'll put the timer back on. So as I think everyone appreciates here, that advanced materials and technologies are ubiquitous in our everyday lives. Probably from the moment you woke up this morning when your mobile phone vibrated or kind of gave an alarm to probably the moment you go to bed when you'll set your alarm again for the next day. Almost everything we do is kind of requires technologies either for banking, for healthcare, for heating, air conditioning, and all sorts of other things. And these obviously all use a variety of different energies and a variety of different materials in order to realise these so that they kind of fit within our daily lives and can therefore be really useful to us. That is odd. Okay. It doesn't change on my screen here, but if it changes on this screen here, then that's all that matters. So, when you're attempting to look for inspiration as to how to make these sorts of materials and technologies, then I think, as most engineers or scientists would, I might look towards biology, because biology has evolved a variety of different ways of doing really interesting things with energy. If you are if you're a fetus rather, inside your mother's womb, and even Donald Trump was one of these at some point, then there was an electrical potential across the spinal cord which helps serve to organise the nerves, and it helps organise your nervous system, your central and peripheral nervous system, and that's about 100 millivolts a millimetre. If you cut yourself open, and I don't propose you do this now because I don't have a plaster with me, then your body will generate a potential, and that potential is around 150 millivolts a millimetre. And what this helps to do is encourage the migration of macrophages and fibroblasts to the surface of the wound, where they can then produce collagen, clean up the wound, and do other things. Electric eels and other fish have evolved in order to be able to immobilise their prey using electricity. And so they can generate up to 860 volts and an amp, which is enough to paralyse their prey, and that makes it much easier to eat. And then if there are a variety of different organisms that if they were to sever a limb, then there's a mechanism, a biological mechanism, which helps to regenerate fully functional limbs in the case of the axolotl or the salamanders. And in the frogs, it generates a spike, which is function-free collagen. One of Saba's academic heroes working at Tufts University works specifically on this. And he's probably one of the leading experts in this area. You can also find evidence for the application of light in nature. So the kind of organisms which use light either do it to signal to partners, hey, I'm here, come and mate with me, or they do it as, hey, I'm here, be terrified because I'm going to eat you, stay away. And then you can also find applications where you use magnetism in nature. So inside bacteria, the skulls of birds, and also the skulls of fish, you find aligned arrays of magnetic nanoparticles. And these are really useful because they help the organism to navigate around the Earth's magnetic fields. So as you can probably tell, I'm from England, and one of the very weird sports that we have, sports if you like, not football because we're not very good at football, is training pigeons to go from one location to another. And so there's a competitive sport where you can train your pigeon to fly between two different locations purely based upon their ability for magnetoception. And that is kind of probably only ever going to be popular in the UK. And then you can also look then for how you might use electromagnetism in the real world. So in the hospital, it's used on a routine basis. Most wavelengths of electromagnetic radiation are used for imaging. And so you can find a technology which is underpinned by energy at some different wavelength. You can also find in some cases, as it becomes more ionizing, it might be more useful then for sterilizing your equipment, or perhaps irradiating a site of a tumor and then enabling you to carry out photodynamic therapy. And there's a lot of other applications which are under development at the moment. So again, dental fillings and things like this, which also use light. And this is kind of relatively routine. And so what my research group does is kind of utilize each of these different kind of energies, kind of each of these different forces in some way to develop new materials. So when you look at what's been developed in terms of materials which are electronic interfaces for the body, the first example which was developed was in Sweden, and these are cardiac pacemakers, so electrical interfaces directly for the heart to make it beat at the right, or to make bits of the heart beat at the right rate. There are electrodes which are embedded in the brain and elsewhere in the body, but there are three which are clinically translated for the brain, all clinically translated in order to switch off shaking symptoms. So for essential tremor, Parkinson's and dystonia. Then you've got bionic eyes, so they are being developed. The most exciting versions of these are the Argus 2, being developed by a second site just outside of Los Angeles. And then you've got bionic ears, which are being developed in Switzerland, Australia, and a few other places as well. And in each of these cases, these all use metals. So these all use metals as your electronic interface for the body. And in each of that, what that means is that the body recognizes that you've just stabbed it with something, and the fact that the mechanical properties of those metals are radically different from the mechanical properties of the tissue which are implanted means that your body recognizes that, and there is then an immune response. And so what happens is then the metal interface gets coated with a layer of insulator, not too dissimilar from this. And what that means is then that when you want to deliver a therapeutic potential to the surface or to the interface, then as the layer of scar tissue builds up, you've got to deliver sequentially higher voltages in order to have that therapeutic effect. And that can then generate tissue degeneration, because you might get to potentials where you split water into, you form radicals, and if you form radicals, you can accelerate the formation of amyloids and other tissue degradation issues. Something which is under clinical trials at the moment is actually not metals, but it's condoms where they've got graphene in the condom. And I assume this is going to be conductive for extra pleasure. So you should see a few other examples of those under clinical trials in the foreseeable future. So what we're doing is taking a materials agnostic and collaborative approach to doing our research. I think it's important to be materials agnostic because there's lots of companies out there who are developing electronic interfaces for the body, which are metals or alloys or kind of inorganic substrates or inorganic materials. And then there's opportunities for a chemist like me to come in and say, maybe we can do something to help. Maybe we can make polymers which are conductive and softer, so it might fit the biological niche where you're going to implant it. Or perhaps we can make things which are degradable. And that opens up a completely different application space. We also want to kind of optimize and solve problems with technologies which are on the market already. And those could be biosensors, those could be electrodes which are implanted in various different places. And then, as I've said, you can develop organic electronic materials which are going to be conductive. And so then we will attempt to make them kind of biologically active, so that maybe there will be more cell adhesive, that the body would recognize it as not being something which is obviously foreign. You might make it so that it was biodegradable, so that in principle you can open up for drug delivery. You might want to make it so that the mechanical properties are analogous, so that if it was going to be implanted in the brain or elsewhere, the mechanical properties are of the order of kilopascal, the kilopascal regime. So attempt to make something with analogous mechanical properties. And then also topographical properties or topological properties, where there are bits of your body where there are aligned cells, or in your bones there may be aligned pores. In your peripheral nerve, if you remove that from your body, then you can wash out the cells, and you see that it's effectively a series of tubes, which is again not too different from the wire here. And so attempting to make materials which have got mechanical, topological, chemical properties which are similar to the places in which they are implanted. And so I'll show you a few examples of these. So there's some of the materials that we would work with which are non-degradable and therefore useful for long-term applications would be polypyrrole, polythiophene derivatives, polyaniline, PDOTs, graphenes and fullerenes. And then if you use a little bit of chemical intuition, then you can generate a variety of different materials where you might have an electroactive block and then flanked either side by something which is not electroactive. So it would be an insulator, but it might be water soluble, so that in principle you can get them to assemble into liposomes or micelles or something like that. Or in the case of polycaprolactone, then polycaprolactones tend to melt around 60 or 70 degrees, so you can melt process them, you can electrospin them, you can do all sorts of other things where you can you can kind of capitalize upon their properties of being relatively straightforward to process. And then these would all be for long-term applications, so for neural interfaces. And on the right-hand side, it would then be either for use as tissue scaffolds for tissue engineering, or alternatively for drug delivery devices. And so we take broadly a biotechnology informed approach to doing this. So a number of my research group look at sources of materials which are biogenic. So on the previous slide I showed you, graphenes, conducting polymers are all made from oil, all made from fossil fuel derived oils. And what we're attempting to do is find biogenic sources of materials that we can incorporate in these, which will be conductive. And so I'll show a few examples of those later on. So the reasons that we would want to do this are that they would be renewable, that they would in principle be lower cost, and there's kind of a lot of other, maybe you can process them using water. So in the long term, there's lots of interesting opportunities. We can dig into marine resources, into agriculture. I've got one of my PhD students who's looking at waste, food waste. I've got another one who's looking in the ocean for sea eggs and other things which have got conducting polymers embedded inside them. We do a little bit of ethics in collaboration with folks in the outside of the chemistry department. We're looking at using enzymes, so we have industrially relevant processes, and also then looking at foods and insects as other sources of these. What differentiates us from what everyone else is active in the area is that we have a kind of a filter, and so that's a gold biotechnology filter, where we use computational approaches to assess how toxic and or biologically biocompatible these things might be. And so there's huge databases of information out there which offer you the opportunity to screen against known toxicity. So whatever's reported in the scientific literature is uploaded into these databases. And then the top 20 pharma companies in the world are also buying into this. So GSK, which is also located on this campus, and kind of Pfizer, Syngenta, and other companies which are making low molecular weight organic species typically for drug applications or for agroscience applications. And then we're kind of adapting this for our own use. And so we use this as a filter before we take it on to the next step, where we make something new. We will look for an application for this. We will make a material which might be very conductive or it might be degradable. It will be useful in some other way, shape or form. And most of our interest is directed towards medical technologies, but in that space. And then if we can take that on and make something really useful, we hope to translate it. And I've got my first PhD student and now postdoc who's taking some of the technology he developed through the Lean Launchpad program, which is a pre-accelerator stage before the startup. So when I've said we're bioprospecting for electronics, there's loads of biomass out there in the world. So there's lots of food waste that we can mine, where we can, in principle, we would be looking for things which would probably be coloured. And so when you look inside the audience members here, you've got some folks with blonde hair, some folks like ourselves with a bit of white in our beard. We don't have very much melanin in our hair, okay? But the folks here with brown or dark or black hair have got a polymer which is called eumelanin, and that's a long polymer. It's conjugated, and the reason it's black is that it's a broadband absorber, because you've got lots of conjugated bonds, and when the molecular weight of them is high enough, then they kind of absorb in the kind of near infrared. Alternatively, there's one person here in the audience who has a little bit of red in their hair, or pink. In principle, if it was orange, my beard used to have some orange or red inside here, but obviously with age that has disappeared. The reason that the orange was there is because you've got an aromatic unit which is a thiophene, and when you have short chains of thiophenes or long chains of thiophenes, you get oranges and then reds. And again, you can look into biomass as sources of this. I've got actually one of my first PhD students looked inside blood, because the heme, which is the oxygen binding unit inside your blood, is also conductive. It's a short oligomer of pyrrole. And then looking for other sources of these pyrroles inside nature. My first postdoc I tortured with a database which had been generated by one of the research councils, which was looking at, it's called BerryBase. And it's all of the products which have been produced by berries to try and find things which would be optically active and also conductive. And he screened 760 compounds for their conductivity and biocompatibility. And that's only a hint of some of the torture that I'll expose you to across here. But anyway, we're beginning to mine different sources of these. And I'll show you one or two examples. Again, the reason we're doing this is because it's green sources, they're processable, probably in solvents which will be biocompatible, so water, maybe some ionic liquids or supercritical carbon dioxide. And again, low energy processes instead of the super high vacuums that you might see elsewhere. So some examples of what we're doing is kind of delving into the melanins. So some of the examples I've talked to you about already. The bright colours here tend to be melanins, so the reds, the oranges, the blacks, they tend to be melanins, and they tend to be polymers of these different aromatic units which are here. One of my PhD students is looking at a medical condition, so she shared between my group and a group in the med school, where they are looking at a medical condition called alkaptonuria, which is more commonly known as black urine disease, because the first time you'll be diagnosed is when you go to the toilet and your pee's black. And then you'll be scratching your head thinking, what on earth? And it was only through having a coffee with a friend that I found out about this. When she said, oh, I'm doing some ramen today on urine. And I was like, why on earth are you doing ramen on urine? Well, it's like, it's black and it might be interesting. And when I heard it was black, I was like, oh, that's probably a polymer inside there. And so that's when that kind of project spurred out of kind of being just a bit of a random coffee one day. And so the polymers here are made of this homogeneic acid. There's an enzyme deficiency in patients with this condition, and they don't process that aromatic enzyme, and therefore its concentration builds up inside your body, and then you get weird depositions of colour. So patients might have black spots in their eyes, blue ears, kind of blue ankles, all sorts of other weird and wonderful colours, which are generated by a build-up of this sort of conducting polymer. So we've shown for the first time that these really are conductive in some measurements with some collaborators in the Department of Physics, and the first paper on this was out just at the end of last year, where the next few papers are going is in looking at the sorts of complexes that these polymers make with the extracellular matrix, so that we can understand from a supermolecular chemistry perspective why they will deposit in certain tissues and not others. And then somehow I'm also torturing the colleague, well, one of the PhD students and a colleague of mine, is through doing electrochemical microscopy. And so this is an optical microscope image where you have either kind of histochemically stained samples, but you can see here that there are some black-blue particles inside there, and those are those pigment particles I was talking to you about. And then using scanning electrochemical microscopy, we can measure the presence of these, or analyse the presence of these inside tissues, and so that might offer an interesting way of probing the mechanism that underpins why the conducting polymer deposits in specific tissues inside the body. And so from a really fundamental science perspective, that's kind of something that we have an interest in. We're also looking inside lignins. And so lignins are the second most abundant polymer on the face of the earth, cellulose obviously being most abundant. So there are millions of tons of these produced on an annual basis. And when I look at this, I look and say, there's loads of aromatic units in here. We must be able to do something interesting with this, particularly when I started and my startup package was £0. It was a cup of tea and a handshake. So I looked at this and was like, yeah, maybe I can do something interesting with this. And so I found some sources of lignins, and then we used some very simple chemistry in order to make these conductive. And then in collaboration with some folks in the University of Strasbourg, where I used to work, then we're looking at how we might be able to use these for energy storage applications, either as capacitors or perhaps battery applications. I've got more interesting things I'd like to do with them, but I don't have the apparatus to do those in Lancaster, and so you have to kind of collaborate where that's not possible. And so with ARTA, then we're looking at how you can charge and discharge these in a kind of controllable fashion. The IP for this is disclosed, but it's not yet granted, and so that's why I can't talk about this in a little bit more detail. But at some point, I would be very happy to. He's prodding me now to say we need to publish, but I think we can hold off until we have a patent in place. And so what I'm going to show you now is examples of how we can apply these sorts of materials. Not all of these are going to contain the biogenic biomass that I've shown you to begin with, but some examples where we do neuromodulation, some examples where we do tissue engineering, and some examples where we do drug delivery. And as I said, the key in all of this is before we start to do this in silico biocompatibility prediction, so that we can then say, I've made this, it's kind of interesting, can I really stick it inside with someone for a long period of time? Or is it something which will only be able to go in temporarily, have a therapeutic effect, and then be withdrawn? And with some examples where we're doing drug delivery for cancer, with electrochemotherapy, there are devices which I can only describe as devices of torture because there are pins which are about 1 inch in length, which get stabbed into people's cancers, that they then do electroporation of those cancers. And so there's a method or a reason that we do this. There's lots of different systems which are out there. So most of them are based upon data which has been uploaded to the scientific literature. And most of them are things that you can then mine there's some open resources. So the ERC has got some open resources so that you can, in principle, look at this. But I think the key is we're using a specific piece of software or specific family of software, Derek Nexus, Sarah Nexus, and Vitic, and it's from a startup company based in Leeds in the UK. And what's really handy about this is that it's affordable. So as a relatively early career member of staff, I'm not paying hundreds of thousands of pounds for access to it. And it gives us really, really interesting insight into, is the material carcinogenic? Is it teratogenic? Is it mutagenic? Is it a skin sensitizer? So when people want to make transdermal devices, transcranial, other electronic interfaces that would work externally to the body, are these going to be biocompatible for a long period of time? And then if you were to implant them, again, you'll answer the same sort of question. So some really, really simple examples of where we would start would be to print a multi-electrode array and then backfill. This is not a project which went well, I'll be very honest. This is a project which was supposed to go very well. It was supposed to be all organic and all degradable, and we got about maybe 9 months in because it was only a 1-year project, and then I had to have some students who would wrap it up at the end. We tried with graphene, we tried with nanotubes, and in the end we tried with silver, and the silver was the only thing that worked really well. Graphene, you could sort of inject it, you could sort of get something that would pop out the other end, and the nanotubes again, the nanotubes are a nightmare to inject and a nightmare to process into these predefined pores. But this was just a kind of proof of concept of where we were trying to go with this. I think maybe in 5 or 10 years, maybe 5 years, when we've solved the problem of how we polymerize these biogenic units inside the pores, then we will have something which is all biodegradable and something which would be cheap and disposable. So these cost about £3, so it's about €5 or thereabouts. And so from a lab-based bio kind of perspective, that's pretty inexpensive because the commercial alternatives to this are hundreds of euros each. And so if you can have something and then you can subsequently dispose of it, that's quite useful. But what I'll say the bulk of the last 5 years has been focused on is doing interesting things with 3D printing, or more interesting things with 3D printing, where we have so a piece of kit which is called multi-photon fabrication. And so here you've got 2 laser beams. The laser beams are in the near infrared. And so if they're in the near infrared, then they can penetrate things which are transparent. Okay? So you can take a PDMS substrate, and because it's transparent, the laser beams can enter the PDMS. And so in principle, you can print into the PDMS, you can print a contact pad on the top of the PDMS, and then through and up at the other end. And so you can have a wire which goes through. In principle, you can print a multi-electrode array in this way. And so this is a PDMS substrate where we've printed polypropyl wires in here. We've got PDOT, we can do the same with PDOT as well. And we can print these over quite large length scales. So here it's about a centimetre. Contact pads around 2 millimetres or thereabout, or can be up to around 2 millimetres. But in this case, it's about 100 microns. In terms of thinking about what size ranges we can print down to, so this is a stress test to ask what the smallest size feature is. And at the moment, the smallest size feature is approximately 6 micrometers. And so from a biological perspective, that's quite useful because cells tend to be 10, 50, 100 microns in size. So you can print some really interesting features which are genuinely useful from a biological perspective. PDMS is relatively simple to print inside because it's transparent and flexible. And so just to see how far we can push that, then we looked at printing inside shape memory polymers, where the idea behind that is that the shape memory polymer would have one shape initially, and maybe you want it to wrap around a peripheral nerve. So you could then have your material, it's transparent, you print your electronic circuits inside, you print the contact pads on top, and then a surgeon can deliver it to a point in the body where it would then unfold or fold around the piece of tissue it's supposed to be in contact with. And so the sorts of things that we can print are prototype wires and something which looks like an I love chemistry. And that's really, really early. I tortured a master's student to do that. And he's just about to finish with his PhD, maybe in a few weeks' time. To take it a little bit farther, then we tried inside decellularized tissues. And this was a sciatic nerve, so extracted from the hind limb of a rat, where we washed the cells out. Okay, and so these are translucent, so they're not really transparent, but when you've got the monomers and the other ink inside there, then they are more or less transparent. And so you can begin to print features inside these. And so these are the sorts of star-like structures you can print inside there. And again then, to try and push that a little bit farther forward, this is inside a plant, this is inside a live plant, where we've printed, because the plant is transparent again, or transparent enough, we can begin to print wire-like structures inside the plant. And you can see here the cells which are from an onion, because I really do like to make my students cry, because this was seemed like a sensible place to start. But here you can see prototype wires that we're beginning to print inside these plants. Then to push it a little bit farther forward, to ask what would be more complex, is then think about printing inside something with blood, because then it's a biological chromophore, which is going to complicate the printing process, because it will interfere with the lasers. And so here is the same wire that we saw on the previous slide, but this time embedded inside a piece of chicken breast. Chicken breast is great because I don't need ethical approval to use it, because I can go to SPA, which is maybe a 2-minute walk from the chemistry department, and pick this up, slice it up, and then do some tests. But this demonstrates that we can, in principle, print inside a piece of chicken, or a human, or some other more complex organism. And we can also electrically characterize it and demonstrate that these are actually a little bit more conductive. I think partly the reason they're more conductive is because here we've changed the dopant and we're using a polymeric dopant. So in this case it's using hyaluronic acid, which is a component of the extracellular matrix, and that helps to keep the polymers kind of rigid and unfolded, and therefore more conductive. And then this is an example we've printed inside a living organism. And so the organism is alive both before and after the printing process. So this is C. elegans, it's a transparent nematode worm, and here we have examples of a star which will be printed inside these, or the I Love Chemistry again, printed inside the C. elegans. And I think again that's really neat. So there's only a couple of other examples out there where people have printed biomaterials directly inside a living organism, and those are all for non-responsive, non-stimuli responsive biomaterials. So they might be for regeneration of soft tissues or something like this. But this is in principle a demonstration that we can begin to print electronic circuitry directly inside a living organism. And so that's where this project is taking it farther forward. I've also got some electrophysiology data for these. So this, which I'm surprised I didn't show you. So after we printed, this is inside the PDMS, sorry, I should have shown you this. This is optical coherence tomography to demonstrate, so we can quantify the print fidelity of the structures we're making. And so this is a CAD file for the desired structure. These are then the OCT images of the structures that we are printing. In the X and Y dimensions, we print with about 100 plus or minus 5% of fidelity. And in the Z, that's much more challenging because the lasers begin to encounter dyes and other things. And so the variability there is much greater. It's up to about 500%, depending on how thick you want to make it. This is scanning electrochemical microscopy images. So this is a millimeter-sized pad, which is conductive, a PDMS substrate, the wire is printed through here, and then here we've got an electrode array at the other end. And this is just to demonstrate that we can use these for to stimulate a piece of tissue in the brain. And so here we've taken these down to University College London, to some folks in the neuroscience department down there, and demonstrated that in principle we can stimulate a piece of brain tissue using the materials where we've printed inside the PDMS. And now we'll go back to where I thought I was going to be, with what else can we do with these? If we can print something which is a dye, you might be able to make optically readable elements from the outside. And so this is PDMS, where we've got a QR code which has been printed inside these materials. And so if anyone was brave enough, if you want to have a QR code reader on your mobile phone, you're more than welcome to come up and try and see if that works. It does work in the lab. I've not yet tried it on a screen like this. This was obviously an early prototype of a QR code, and this one is actually the QR code which would read out to the university's website. And so we can print with quite a decent degree of fidelity, and QR codes are obviously kind of very comforting because you can have some defects in there and they'll still work to a greater extent. But we've moved on quite a long way from where we are with this now, not only to printing inside PDMS. And then I think it's also interesting that, or it's worthwhile explaining what we do to people outside of the university, because if you want to inspire the next generation of researchers, then something as simple as a smiley face is probably a useful thing to have. And again, that's conducting polymer structures which have been printed directly inside these. And so I think that kind of draws what we do with 3D printing broadly to an end. And then I'll show you some stuff with tissue engineering. And then in about 3 or 4 minutes time, I'll show you some examples with drug delivery. So if you have a conductive matrix, you know from the fact that there are electrodes which stimulate the heart and the brain, that maybe control your bladder function and do other interesting things, that perhaps you can stimulate cells to do other interesting things. One example of this is your peripheral nerves. And so if you have a massive nerve injury, so if it's 2 centimeters or more, then what normally happens is the body backfills the space with scar tissue, and then it doesn't function. And so you normally lose that. What people then do is try and generate biomaterials which will have pores so that they will help the nerve regrow in the same way. In some cases, they might try and make them conductive. And what we're trying to do here is take a conductive scaffold, pop some Schwann cells on the surface, which Schwann cells normally kind of coat the outside of the nerves, and they generate myelin. And by making these scaffolds, we can then grow the Schwann cells on the surface, electrically stimulate them, and then they will produce nerve growth factor, and that helps to heal the nerve injury much faster. We can do some other interesting things where these are more directed towards other parts, say the brain or elsewhere in the nervous system. Then you can make a conductive substrate, and then what you can then do is plate cells on there and look at the number of neurites which outgrow from these. And these are cancer cell lines where the differentiated potential is estimated in terms of the neurite outgrowth length. And so you can see your positive controls and negative controls. And what you would typically see then is that after a specific period of time in culture, you can have greater amounts of neurite outgrowth and neurite length if you are on a conductive substrate. We also look at making architectures where we electrospin something. We can make sure that those fibres are all aligned, and if the fibres are aligned, then when cells interact with them, then they will tend to align with them. It's not compulsory, but in some cases they do. And so in this case, we have examples of silk, which has P dot and PSS, which is grown inside there. It's coated with laminin, and then we do mechanical tests to demonstrate that the mechanical properties are approximately of the right order of magnitude to be used in the central nervous system. And then we would look at how neurites would outgrow. And in the cases where the fibres are aligned, then you can see increased neurite alignment on those substrates. And so that's a method of, so the electrospinning there is a method of making materials with topographical guidance cues to ensure that the cells behave in the way that you want them. And then we've also got other examples where we've made, again, conductive materials, where we might plate muscle cells on the surface. So again, these are from the cardiac tissue, so mouse atrial fibroblasts, myocytes rather, and then look at how cell organization changes and look for the formation of gap junctions on those, and demonstrate that when you have a conductive substrate, then the relative conductivity, so the formation of the gap junctions, is increased relative to those on the non-conductive substrate. So there's some interesting kind of biochemistry which underpins why that actually happens. And then I guess the last sample I'll show you with tissue engineering for is then for printable so polysaccharide and polypyrrole PSS gels, where we've used these here for cartilage tissue. And you can kind of print so these is in collaboration with some folks in Germany, where you can print a substrate, you can then render it conductive using really simple chemistry, and then grow cells on the surface of this. So this is your kind of standard tissue culture plastic. Cells don't really grow very far into this. Where you have the gel, which is non-conductive, you see that the cells do begin to migrate in. And then when we have the conductive substrate, then they migrate a little bit farther in. And the reason for this is partly A, because we've exposed the matrix to acid, so that's going to degrade some of the matrix and make it a little bit more porous and a little bit more swollen. And then you see that cells grow quite happily on the surface of those. And I'll show you a handful of examples where we've done it for bone tissue. Here, when we generated the first examples of conductive materials, which were then biomineralized. And so your bone is obviously a complex mixture of vascularization and soft tissue and then the hard matrix, so collagen and hydroxyapatite. And in order to make these materials so that they would look a little bit more like what you would expect to see in vivo, then you take polycaprolactone, you can render it conductive, and then if the conducting polymer displays amines on the surface, you can use those as sites for nucleation for silica, which is the sort of thing you find in marine organisms. Or if you have carboxylic acids on the surface, you can deposit calcium phosphate, calcium carbonate, which are the sorts of things you'd expect to see inside human bones. Then you pop stem cells on there and electrically stimulate them, and you can measure the upregulation of the production of alkaline phosphatase activity, which is a hallmark for healthy bone formation. An alternative to this would be to electrospin materials. Again, polycaprolactone is the electrospun substrate, grow an interpenetrating network of the polypyrrole PSS, and then plate human mesenchymal stem cells on there and electrically stimulate them and demonstrate that you can upregulate alkaline phosphatase production and activity, and also calcium deposition. And again, those are hallmarks of the onset of healthy bone formation. And a number of years ago, this is an example where we had a foam, so where the foam had macropores inside it, and the macropores are 150 to 400 microns in size. And what that means is that then it's easy for vascularization to regrow. And if it's easy for vascularization to regrow, that's probably one of the challenges, I would say, in macroscopic tissue engineering at the moment. And this was an example where we took a protein, so silk from Bombyx mori silkworms. We made foams from that, made it conductive, and then put stem cells on there and electrically stimulated stem cells, and then drove their differentiation towards an osteogenic outcome. And at the time when this was published, this was by far and away the largest example in the literature, at a cylinders of 4 millimeters in diameter and about 6 millimeters in length. And so enough of the tissue engineering. And as I said, when you see Grandpa Simpson, then we're coming towards the end. So the reason I think Grandpa Simpson is a really good kind of figure to have on here is because it will tell you about the market, the reason, the kind of driving force that we're doing this. So I've got folks from Breaking Bad on here because they're chemists doing something which may or may not be profitable and may or may not be legal, but they're doing something which is using chemistry, and this is capturing the public imagination. You've also got then Frankenstein's monster. Okay, I've shown you a few examples where we had electrodes earlier on, and then the characters on the right are a baby who's about to be injected with something, or Grandpa Simpson. And so problems you might find with typical drug delivery devices are patient compliance being one really huge one, where if you've got babies, so typical examples of drug delivery system would be a tablet or an injection or cream. And in each of these cases, the profile of drug delivery that you would get is a burst, which would kind of peak and then come out to a trough really quickly normally. If it's an injection, you might inject yourself. And so there's not that many people out there who really enjoy sticking needles inside themselves. Although it's plausible with this crowd, maybe there's one or two of you inside there who do enjoy sticking a needle inside yourself. And then Grandpa Simpson is a really good example of aging populations. And so we do have aging populations. And I was at my aunt's 90th birthday 3 years ago now. And in between drinking and celebrating the fact she got to 90 years old, we also went into the kitchen where she kept cakes to go and get some more cakes to kind of go alongside the alcohol. But then when we were there, we also looked inside the drugs cabinet because she had to remember to take her medicine. And she said, oh, yes, of course I've taken my medicine. And on the side, there was a piece of paper, which clearly demonstrated she hadn't. And then we looked inside the cupboard, then there were drugs from 5 or 10 years ago, which are past their use-by date. And so as you have an aging population, there's a definite need and necessity for systems which might facilitate people to take drugs. It's not only useful for aging populations, but also people with memory conditions, people with a lot of other kind of reasons why they might forget or just not want to take their drug. And so I'll show you a handful of examples where these drug delivery systems are all electrically controlled. And if they're not electrically controlled, then you can use light or magnets to do that. So I won't show you the material, but in this case it's silk loaded with reduced graphene oxide. And then on the surface of that, we have nerve growth factor beta, which is a biologically active part of nerve growth factor. Obviously, there'll be some desorption of the nerve growth factor from the materials, and that's relatively low, about 10% of the loading over the period of about 10 days. If you were to use electrical stimulation, you apply potential to the film, that acts as a pressure in order to make the nerve growth factor desorb from the surface of that material. And so over the period of, say, 10 days, you can control and you can switch on and off the delivery of that macroscopic drug, that high molecular weight drug. And so that's quite an interesting way of getting around the issue of patient compliance, because in principle, this demonstrates really clearly how much control you can have over the delivery of a drug in response to that electrical signal. The example I'm going to show you on the right is an example to demonstrate I don't have a clue. Right, what this shows you is that this is a similar system, except the polymers which we use to make this are different. And so instead of using graphene and protein, we have here a polysaccharide and polypyrrole. And this is the same drug delivery paradigm, trying to deliver nerve growth factor. And we show that over a shorter period of time, we don't have anywhere near the same level of control. And so we're collaborating with some folks who do computational modelling to understand why we don't have that same level of control, and to look at the supramolecular interactions between the proteins or the macroscopic drug and the drug matrix. So that does definitely highlight that that's important. And here, then we have another example where we have a protein loaded with a conductive polymer, and then we're delivering gentamicin, which is a broad spectrum antimicrobial agent. And again, you can see that we can have passive delivery, so this is the leaching, which is normal from these materials, and then when you apply an electrical stimulus, in each case you can really switch on the drug delivery in response to that paradigm. An alternative example would here be a polysaccharide loaded with the conductive polymer, where we can deliver, in this case, pemetrexed, which is an anti-cancer agent. And how we're taking this farther forward is in collaboration with a cancer specialist in the hospital. He uses devices which I can only describe as torture devices, which prod the cancers, and they electroporate the cancer. And when they electroporate the cancer, in principle it dies. But if you coat those pins with this sort of material, in principle you can do two things. You can ideally kill as many cancer cells as possible with the electroporation, but also you can deliver an anti-cancer drug which may have greater therapeutic effect. So that's kind of interesting, but what I think is more interesting is trying to do things wirelessly, because it's great to control things using a wire, because you could contemplate easily coating the surface of an electrode that you might put in the brain, cardiac, elsewhere. But these are examples where you can have particulates. So in this case, it's a polypyrrole particle. It's loaded with an anti-cancer agent, so allicin. And then we use, because it's a broadband absorber, you can dump energy in using near infrared, and that will then kick out the drug from that polymer matrix. And here we've used it as an in vitro assay to deliver to HepG2 cells. And that's my time, which is up. So I've got 2 more slides, don't worry. But it does mean you're going to get out for coffee in no time. And so what we can show you here is we can wirelessly trigger the delivery of the drug. And then to take this further forward, here's a radio controlled system or magnetically controlled system where we can do the same from a film which is located on the surface of an electrode. And then this is something I think which is kind of interesting, where we've made examples which are inherently degradable, where you have an electroactive unit, you have a non-electroactive unit, so PEG or polycaprolactone. As I said before, the PEGs are water soluble, the polycaprolactones aren't. And so you can 3D print those instead. Here we've used the same electrical simulation paradigm, switch on, switch off. And this is a clinically relevant anti-inflammatory, which is used really routinely. So it's dexamethasone phosphate. And this was one of the first examples at the time when we published this. And how I've got one of my students to take this farther forward is so these are building blocks from blood. So this is hemin, and these are enzymatic degradation byproducts, bilirubin and biliverdin. Again, they're also electroactive, biliverdin more so than bilirubin because it's more conjugated. And then we've incorporated those in the backbone of the polymers so that then we have something which is biodegradable and biocompatible by virtue of the fact that it's biologically sourced. They're not perfect, I'll be honest, but they are significantly, they're orders of magnitude better than what we were using before. So if you compare it to the oligoanilines, those are short, aromatic and cationic, and so those will intercalate in DNA and be carcinogenic in the long term. And we've eradicated that problem from this because of the in silico toxicity screening. And again, then we've done the same electrical stimulation paradigm to show that we can switch on and off the delivery of the drug. And as a final slide in terms of applications, then these structures, these I can't show you, but these are polymers which contain units where you can either electrically stimulate them or use light in order to kick the drug out. And again, you can do that using so UV visible spectrum light in order to do that. And then this is kind of switching on and off the delivery of the drug. And as I recognise that light can only penetrate so far in tissue, then we've also looked to make things which respond to magnetic fields, because magnetic fields in principle can penetrate all the way through you. And so this is an example where we have a kind of a composite material. We have a magnetostrictive, so it changes shape when you expose it to a magnetic field. On top of that sandwich is a piezoelectric. So when you stress the magnetostrictive, that deforms the piezoelectric. The piezoelectric then generates an electric field, and then it's embedded inside a polymer matrix. And so the polymer then is activated by that electric field and then delivers a drug in response to that. And so that's all the kind of weird and wonderful examples I'm going to show you over the course of today. Lots of these people are the people who I've got to thank for stuff which is not yet going to be kind of ready to talk about for another year or so. But the postdocs are here on the left. So 3 from Turkey doing something synthesis, architecture and biology. Mark's doing the startup thing. James is working on the 3D printing in vivo, and this postdoc is actually working in the management school, so doing something completely different. And then the PhD students and the master students on the right-hand side who are doing the kind of more adventurous stuff. I've obviously got to say thank you to collaborators across Europe and elsewhere, and if you have, and obviously to the funders who make this actually possible, because without them I wouldn't have the opportunity to be here with you today. So if you have any questions, please don't hesitate to ask.
Thank you very much, John, for the very good presentation, very interesting. So, is there any questions in the audience? No questions? Yeah.
I have a question. When you were printing inside the tissues,
Yep.
then to test them, you have to really go with cables there and...
So yeah, the electrical measurements we did inside the piece of chicken breast were 2-point probe measurements.
But the aim is to make a complete circuitry with this printing.
Yeah, yeah. So I'll show you where I'll give you a hint as to where we are. So this is a QR code which we can do with relative ease. So we can begin to make structures which are like indigitated capacitors.
Okay.
That's about where we are at the moment. But the challenge, what we'd like to try and do, is have kind of 2 layers of structures. But it's really difficult. It's not. I mean, this was not easy, right? This is I think 4 postdocs, 3 master students, and 2 PhD students to have got this far, right? So it's been a slog. But we're now at the point where we can reproducibly print wires and things inside there. The next, and that's in a 2-dimensional. When you move to the 3rd dimension, I've got so James, what James is doing at the moment is printing a series of layers. So 1 pad, and then another pad above it, another pad above it. So we're looking at what distance we need to be away from one another to make sure that the ink doesn't bleed. Because when you're doing these polymerizations, you heat things up, it's quite messy. So it's not like vacuum deposition, which is beautiful and gives you something nice and reproducible almost every time. It's because you're doing something in a biological system, it's much more tricky. And so I think it's about 12 microns, which is about the separation that we need between one pad and another pad. But that's a work in progress.
Thank you.
Right.
Other questions? I have, yeah, sure.
I have one question related to the nerve growth factor. When you apply the electrical stimulation to release it, is there any option or alternative to do it in a way to maintain constant concentration of the nerve growth factor instead of increasing the release of the drug?
So hypothetically, the answer to that is yes. So here, this is over a period of hours, and so what we were looking for here, what we were measuring, is the amount of nerve growth factor which is in the cell culture media after a specific period of time. In principle, if you wanted to, it would be possible to have it so that you electrically stimulate and maintain a constant, but that was outside of what money can buy. And so if you have 100,000 euros or something down the back of your pocket, then I'd be more than happy to invest that with you in taking that experiment farther forward. I'd be delighted. You're more than welcome to come to the lab. I'll show you how we hook up the systems, and then you can come back here and work on something like that with Saba.
Any other questions? I have one question myself. So could you a little bit elaborate on printing inside the transparent construct? So how you insert the material inside, basically? So because you talked a little bit about the laser penetratable to the system, but how you deliver the ink?
So let's take. So all of these are simple example. Let's. I don't know which one's. Probably this one's the easiest one to visualize. Okay. So you can see the fact that this is shiny, and this is a thin layer of PDMS. So it's about somewhere between 4 and 12 microns. But it's been deposited by spin coating. Okay, so you get a controlled layer of PDMS, and then if you want to print something like this, you just apply some ink, and the ink, because it's aromatic and it's kind of an organic thing, it's going to swell the PDMS chains. Okay, so the PDMS will swell to a certain extent. I've got a computer modeler who's looking at how we can kind of control the 3D printing process in the long term. But what we are, so in principle, your polymer matrix is transparent, so it's optically transparent, and the ink swells the polymer matrix. And then the lasers can penetrate that swollen polymer matrix and print inside it. You can do that again with things which are not perfectly transparent. And so these are not perfectly transparent. This is a shape memory polymer, and it's not perfectly transparent, but it's transparent enough that you can print inside there. And so you can print quite large structures. These are maybe 100 microns or something like that. And the wires here are, so this is about 1 millimeter. And the thickness is then around 100 microns, maybe 50 microns, something like that. It's not. This is by far and away. It's not perfect because it was a master student who was doing it, and we had kind of limited time and resources. And here again, this is more complex again because you've got objects here. So your extra kind of bundles of ECM, which are kind of the size that they will begin to scatter light. And those make it much more complicated. And what we sometimes saw was ablation of the substrate. So we saw ablation of the matrix. And we've also seen that when we were printing on ITO, that we might kind of ablate the ITO if we don't focus the laser beam in the right place. And then with the plants, those are really, really tricky. But it's not impossible to print inside there. It's just their cells are much more robust than mammalian cells. And so when I show you these are much more straightforward to print inside, despite the fact that they are coloured, and that would complicate the kind of ingress of the lasers.
Thank you, John. So that's it, I guess. Thank you very much.
You're welcome.
For
Cool.